And introduce the next speaker, Jennifer Mcclure from ICBF, the Irish Cattle Breeding Federation, and she's presenting a paper on genetic defects in collaboration with Chagas and the University of Missouri.
Good afternoon. I'm going to talk to you a little bit about the prevalence and discovery of genetic defects in the national Irish cattle population. So there we are. In case you are not familiar with the Irish demographics, there's about 128,000 farms. The average farm size is about 32.3 hectares. We have 6.4 million cattle and 3.3 million sheep, which means that if you were to hand out a cow per person, you'd still have a few left over in this country. We have had a 3% growth in our agri-food exports in 2015, meaning agriculture is a very important part of the Irish economy. It brings in $10.8 billion every year. 12.3% of our exports are from agri-foods, and it employs 8.6% of our population, 8.6%. We happen to be the #1 beef exporter in the EU, although with Brexit, I'm not sure if we'll still get that, but we're not going to talk about that. We are #4 in the world as it sits, and we provide 15% of the infant formula worldwide. So we also have this neat system called AIM, and that's through the department where they track movements of the cattle. So once it's born, they get the birthing records, then they get moved, and we have so far 1 million farm-to-farm moves, 1.7 movements via marts or the auction houses, and all of that was recorded in 2015. So this is a huge database that we get to pull from in terms of that sort of stuff, but we also end up getting a lot of genetic material as well. My PhD is in vet pathobiology, so I'm not a geneticist, but what I'm very interested in is the genetics of health and disease, and specifically one of my pet projects is this genetic defects. We know that all animals carry a number of lethal recessives. In fact, a paper from 2015 mentioned that in humans, 1 in 2 humans carries a lethal recessive. We're a little less inbred than our animals, which is why that number's a bit lower, and they estimate that in animals, it's going to be higher. So we've also found that many animals are carriers for different disease alleles. You may have a particular sire, Oman's known for contributing very, very well to the dairy population in terms of milk, but he also has a number of genetic disease alleles as well. But his contributions outweigh that, so everybody still has them in their pedigree. Each year, new SNPs can be found for these genetic diseases and SNPs that can cause early embryonic death. And so what I wanted to know is how can we facilitate their discovery? So you can't fix something you don't know about. So if I don't hear about these genetic defects, I can't do anything to fix them. So what I did is I borrowed some information from colleagues in France. I have a colleague at ICBF who was very kind and helped translate some things I didn't know what they were. And we create a head-to-tail survey. Within the survey, it covers anything that might go wrong with the animal. Some are genetic, some may not be, but we're starting to get this data into our database. It's a big collaborative effort with farmers, veterinarians, our regional veterinary labs are contributing, and I've even gotten some researchers on board with this as well. What we then do is we then either do whole genome or exome sequencing on a few of these animals. That's one tool where you start looking for SNPs that are involved in these specific diseases. And we also have a tool very useful to us called the International Dairy and Beef IDB SNP chip. Now, this is going to end up sounding a bit like an ad for it because, God bless him, I'm married to the man who helped develop this. So I promise we don't get any money off of it, but we did develop, it was developed at ICBF in conjunction with Chagas and Weatherbees and a few others. It was developed with imputation in mind. It has 55,000 SNPs on it at the moment. We tend to increase every year. It was 15, we're now up to 55. The cool thing is it's got 72 validated disease traits and SNPs. So some of them are beneficial traits, a lot of them are the disease traits, and right now we're sitting at just over 500,000 animals genotyped, and we're on track to do 330,000 animals, Irish and European, every year on this chip. Part of that is because if you buy a whole lot, Illumina likes you a lot, and they give you cheap chips. So By the end of this year, we'll be sitting at about 750,000. I just ran the numbers with the man who did it. And it's a huge population. So we're getting huge, vast amounts of genetic information. This is the list of diseases and traits on the IDB-v3. Good news is you don't have to read this right now because all of the slides will be available and all that kind of stuff when we're done, and along with my random spiel that goes along with this. But there's a couple things to point out. The colors aren't great, but wrong button. There we go. Light blue means that they were newly validated, as in this past year we just validated them. The pink ones require an extra fee due to IP. Some people have put IP on these SNPs, and therefore we cannot give the information away for free like we would to the other ones. Now, a lot of this information comes from the literature. It's been compiled, put together. That's where these SNPs came from. I throw this up because if you happen to visit our website and go to this health and disease page under services, you can look at this. This has a few different important things on it. If you're a veterinarian, you're not familiar with genetics, it has an understanding genetics thing that's great for farmers. It goes very basic. Next is a genetic diseases and traits definition, and that covers most of what was on that last slide, telling you what they mean, what they are. But if you're a geneticist yourself and you want to know where this came from, what it does, where the SNP is, that's in the bottom one. And it also has all the clinical definitions because I basically took all of this and translated it so that our farmers can understand it because they're our end users at the moment. So moving on, let's talk about frequency. So these numbers are very, very small. You're not going to be able to see them, but the point is in the red is anything that's over 1%, and this is in the population of animals that have been genotyped in Ireland. So this is in that 500,000 that we have right now genotyped. To blow it up a little bit, so we have full population, dairy, beef, beef crosses, purebreds, meaning purebred dairy or beef, and then my purebreds specific breeds. Angus, Charolais, Limousin, I chose those because they're the most prevalent breeds, and also Friesian, Holstein, they're about the same thing, and Jersey. And what I want to point out is this is a very good tool for veterinarians because you guys can look at this and talk to your clients and say, all right, I see that in your herd, you've got a few carriers for these specific diseases. So when you're picking out your bull, we need to make sure he's been genomically tested and he is not a carrier as well. Conversely, if you don't have carriers, then you don't have to worry about it. But if the animals are tested, you can then give this advice to the farmers and help them so that they don't see any of these diseases on their farms. So we also have unwanted. Again, these slides will be available afterwards so you can take a better look at it. And this is all very preliminary data. This is working on being published, so it's not out there yet, but it's getting there. But it's showing that we do have some of these things within our population. We also have a lot more breeds than just these ones. I just chose the ones that were most representative. So now I want to tell you my favorite story. This is how I got into this. I got contacted by a farmer, God bless him, and he says, I have a problem with my cattle, they keep falling down. And I said, okay, can we be a little bit more specific here? And the guy said, yeah. One of the guys at the regional veterinary lab, the RVL, suggested that I contact you because he diagnosed progressive ataxia in my cattle. Can you do anything? It's genetic. I said, well, I'm not sure. As an overview, progressive ataxia is a disease that causes incoordination when they're stressed, doesn't manifest until 18 to 24 months of age, typically in Charolais, and it gets worse until they can't rise. So that means that this farmer has put 2 years of feed, 2 years of work, everything into this animal just to turn around, watch it die before he can send it to the factory. He was very upset, and I don't blame him. So he asked me what can be done. So this is my first try. This is a video of the cow. Hopefully, there she goes, so that you can see it. I want to send a big thank you to John Fagan at the RVL. He sent me this video, allowed me to use it for this stuff. There's my farmer. You can see she's going to go a little bit sideways there. The hind end is in coordination. Down here, the more stress she gets, the more uncoordinated she gets. And the worst part is, I think it was about a month after this video was taken, she went down and he lost her. So this is very near and dear to me because it's very upsetting for these farmers to have this happen. So what did we do? Come on. go. There we go. The farmer collected some tissue, hair cards for us. We sent the DNA off to get extracted. We did exome sequencing this time because we found that 85% of disease-causing mutations can be found on the exome. Then I sent it to University of Missouri, to these yahoos, distinguished colleagues, Jerry Taylor and Bob Schnabel, fantastic guys to work with. They're excellent. They ran it through their data pipeline. We identified 10 potential disease-causing SNPs. We're working on testing and validating to find the correct gene if it is on exome. And then we're going to roll out these tests to all the farmers so we can get our Charlet population tested. Let's get rid of it. So recap, 10 SNPs identified. But what we're going to do is we're also going to do whole genome sequencing next, just to make sure. So it's to be sure, and that also means that I get a cute little paper out that says, hey, here's what happens when you do exome and whole genome. Next, testing. We are in the process of testing the sire, the dam, and the half sibs for validation, and then adding it to the chip, our SNP chip for farmer reports. And when I say farmer reports, this is what I mean. Pretty soon, all of our farmers are going to get something like this. When they SNP chip their animals, they're going to get this report back, and it's going to say you're a carrier for this, you have this within your population, again, so they can make these educated decisions when breeding. So, in the meantime, I'm on a hunt. I've been working closely with vets, RVLs, researchers, and these are the ones that I'm targeting next. Ventricular septal defect. I was thrilled. There was a talk on it in Italy recently, yesterday, I believe, and I needed to catch her because if we can get samples from them, that'd be fantastic. Atrasia, we're looking at 3 different types of atrasia because we don't know if it's one SNP, it's multiple SNPs, and that's the other thing. This could be multiple things, but we won't know until we start looking for it. Rhineck, which is this guy here. Dwarfism, cleft palate, cleft nostril, which has actually been linked to one particular sire, and we're seeing if we can find it anywhere else. Schistosomus reflux, progressive ataxia, and also photosensitivity. Now that one got brought on recently because some of our farmers contacted me directly and said, I have a problem, can you help me? So, at this point, I'd like to acknowledge everybody who's helped out with this, and I realized after putting this up, I'm missing a few names. I'm terribly sorry if you're out there and I missed you. I do appreciate everything that everybody's done, and I will happily take any questions if we have time.
Thank you. Any questions? When I look at your first talk, or your first part of your talk, you seem to suggest there are lots of genetic defects, lots of animals have them. So how many do you think there are? And shouldn't be the general advice to farmers not to avoid inbreeding rather than start testing for?
So to answer the first question, how many do we think there are? So we've tested the 300,000, and the most percentage that we saw in a population is the Holstein population for Brachyspinian. I think it was at like 6.8. So that comes out to be a couple thousand max. Okay. So that are carriers for it. But the thing is when you start breeding this, if you're not careful, that 6% is going to become 12%. It's going to be 24. Keep going. So it's an awareness thing. And in terms of reducing inbreeding versus genotyping them, I think that honestly it's better to know what's in there than just guessing, and it's not 100% from the inbreeding as well. Yeah.
But my question is, how many genetic defects you think there are? Because my feeling is that you have a lot on your chip.
Yes.
But my guess would be most of them are not on your chip yet.
Correct.
And that's why I was thinking about maybe we should avoid inbreeding because we take account for all the ones we don't know yet.
Yes. So to answer that then, yes. Definitely, we always want to avoid inbreeding. I mean, we don't want to end up like that. But in terms of how many are out there, I'll just put it this way. I don't think I will ever be out of a job. Because that's the thing with this. When people start breeding very selectively on their breeds, we have this with one particular breed in Ireland right now. I've had 6 different defects pop up from this. And God bless the producers. They want to send it to me and let me know about it, and we're working with them to try and help them breed that out, essentially.
But they should have solved this much earlier and avoid inbreeding.
Yes.
Isn't that it?
But when you have niche populations...
Yes, of course. Yeah, no, I know. We know the similar issue in dogs and a lot of other species.
Exactly.
Okay. No more questions? thank you very much for this excellent presentation.
Thank you.